So should I start?
Yes, please.
Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you all to an exciting lecture organized under Comstech Distinguished Scholars webinar series. We are very excited and grateful to have our guest speaker with us. Our guest speaker for today is Professor Dr. Rana Dajani. Professor Dr. Rana Dajani is Professor of Biology and Biotechnology at Hashmiyat University in Jordan. The title of our talk today is Epigenetics, the Immortal Quest of Our Grandparents. Dr. Rana, we are very grateful for your time, and we are also very grateful to all our participants who are joining us from around the world. And with this, I would request Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS, to introduce and welcome our guest speaker for today. Over to you, sir.
Good morning, good afternoon, and even good evening. Dr. Rana Dajani, you know that your popularity is beyond continent. So we have participants from 4 continents today, and I'm sure that a lot more would be joining us as you will speak. We have had the honor of welcoming you briefly in Karachi, and we're looking forward to have you in Islamabad, this time as a guest of Comstech. As you know, Comstech is an organization which is a premier institution of OIC system responsible of developing science and technology cooperation within 57 states, from Suriname to Brunei Darussalam, all the way to the east, and south to Maldives and Kazakhstan to the north. So spread over 4 continents, there are so many people who are here online, and many more will join us to listen to you. Now, we know you as a scientist, as an activist, as a humanist, and someone with a great insight about the real issues which humanity faces today, and your approach towards solving them, or at least proposing to solve them, is absolutely optimistic. And that is what makes you different from the other, because all the way through, whenever I heard you, and I have been really very, very impressed from the kind of insight you bring and out-of-the-box solutions which you have always proposed. Let me have the honor of introducing a very accomplished lady, someone whose introduction can be spread over the duration of the lecture. But let me just be very brief and tell you that Professor Dr. Rana Dajani, she's a professor of biology and biotechnology at Hashemite University, Jordan. And she is, in fact, a scholar par excellence, has direct contribution in the field of molecular biology and biotechnology. So she has been training people, young people, delivering lectures all over, and addressing issues of higher education, women empowerment, women contribution towards society, and inter-Islamic cooperation. She's a member of the United Nations Women Jordan Advisory Council, recipient of a very prestigious award called Jordan's Order of Al Hussein for distinguished contribution in the field of science and technology. She is a recipient of Library of Congress Literacy Award Best Practices, recipient of A-Stars Impact Award, recipient of Synergos Arab World Social Innovator Award in 2009, and recipient of A-Wise Award. So I'm sure that this list is completely incomplete. And I'm sure that there are so many things which I know about Dr. Dajani, and which actually reflect the stature, the insight, the global vision of a very distinguished scholar. This, I would like to invite Professor Dr. Rana Dajani for her presentation today.
Thank you very much for the invitation and for the introduction, Professor Iqbal. You are also a fantastic scientist and role model and a visionary and leader. And it's a pleasure and honor to actually be part of all the good work that's happening at Comstech. And I always talk about it wherever I go as an exemplar of doing good science and good leadership within the global South. And I applaud you for that. I applaud you for the scholarships you provide to students all over the world. And in any way possible, I am here to support you and all the work that you're doing. So thank you very, very much.
Thank you.
I will start by sharing my screen. Let's just do that. Yes, here we go. Share. Please. Just technology. All right, you can see my screen, I'm assuming. Slideshow. All right, so I'd like to start first by saying Assalamu alaikum wa rahmatullah, peace upon you all. And Bismillah ar-Rahman ar-Rahim. I like to start with this saying, which is seeing what everybody sees, but thinking what no one has thought. This is a saying by a famous physicist Nobel Prize winner. I'm not going to tell you who he is. You know, I'm an educator, so I'm going to make people do some work to check out who actually said this. And his name his first name is Albert, and it's not Albert Einstein. But to me, that's a motto of being a human being. That's how we survived as a species. But it's also the hallmark of a scientist and a social entrepreneur, and actually even your grandmother, right? Making observations and then coming up with questions and then thinking of explanations or solutions. And that's what our culture and religion requires us to do in all and every verse, to think, to reflect, and to really use our minds to the advantage and benefit of our communities and societies and the universe. So with that as an inspiration, I'm going to share, my talk is going to be in a form of a story. Again, I always refer to evolution. We also evolved to be storytellers. That's why all the religious books have stories, because we learn through stories a lot more than somebody telling us do or don't, or sharing a set of instructions or regulations. And so also to celebrate storytelling, I'm going to talk about the story of epigenetics and how I got involved with it. And I really thank Professor Iqbal for the introduction that he gave me, but I want to also share my own little introduction. Is that I like to say that I wear 5 scarves or 5 hats, but I don't wear a hat, I wear a scarf. And the first one is that I'm a mother. And I think that's the most important role I can play in the community and to contribute to society. And we usually don't mention that as a role. We take it for granted, although we should be very proud, because that's the first and foremost thing we can do to contribute. The second one is being an educator, a teacher. I was a school teacher and then a professor at the university. So teaching and pedagogy is the second most important thing we can do to inspire the young generation and help shape and mold their critical thinking skills and their confidence in themselves. So again, second most important role is being a teacher. The third one is being a scientist, and that's what we're going to talk about today. So I won't elaborate, but we do a lot of exciting science that deals with trauma and epigenetics, as well as ethnic populations. And of course, in any work, you don't you remain limited within the confines of your lab or your hall of teaching. You actually go beyond into the community, depending on where the need takes you or the curiosity takes you. So one need that came out of my work as a scientist was actually helping in establishing the first stem cell law to guide using stem cells in research and therapy. So again, going beyond the limits of my expertise and having that light and that fire of learning to continuously burning inside till the last minute of our lives. And so this way I contributed to policy and learned a lot on the way. And this keeps us humble, right? That there's always something we don't know. And the more we know, the more we realize that we don't know. That leads me to my fourth scarf or hat, which is being a social entrepreneur and founding a program called We Love Reading that works on changing mindsets through reading to create change makers. again, going beyond my classroom and lab to solve a problem in the community that I observed, and then use my skills in research and observation to come up with a solution that is local, just like Comstech, and then spread internationally because it's a great solution. And for that, just to share some nice news, we actually yesterday won the Schwab Foundation Social Entrepreneur of the Year 2022 because of the work on We Love Reading. So again, we must reimagine ourselves in not just in one form, but in multiple forms, however the need and the requirements and our curiosity take us. I didn't mention the fifth scarf, and I will not talk about it because there's no time, but this is an invitation to go read about it in my book, 5 Scarves, Doing the Impossible. If we can reverse self-hate, why can't we reimagine or redefine success? So I'll leave you with that and maybe put some links later in the chat for those who are interested to follow up on the book. I want to also celebrate scientists in our community. We always talk about scientists from the West and celebrate them, rightly so. Or we talk about scientists in the past from our culture and heritage, and rightly so. But I think today we are in need of celebrating the scientists who are living with us, or at least within our modern day, so that they can inspire us and be true role models in the sense that we can see ourselves in them and be inspired to think big. So I always start my lectures by mentioning a few scientists who I have encountered, who have inspired me or become role models. And I urge every person, when you talk, give a talk, draw from your own community, your own circle, who are the scientists that inspire you, and share their names so that the world can know about them. And that's how the names become commonplace at the dinner table. So I'm going to share a few. This is Professor Dajani. He's actually my father. He passed away 2 years ago, but he was my inspiration. He was an immunologist, allergist, and he inspired me since I was young to ask those critical questions and to follow my curiosity wherever it goes. Another one is Professor Musa Nazar, who is a hallmark in organic chemistry. Also, he's still alive and an inspiration as a teacher, as a world-class scientist. And then for Professor Abir al-Bawab, who is a professor of chemistry at the University of Jordan, who recently won an award as a distinguished woman in chemistry and chemical engineering, who's also a very distinguished professor and a role model. And lastly, Dr. Suhad Yassin from Iraq, who's also an inspiration and was featured on Vogue to change how people look at women as scientists, not just as in the typical stereotype. So with that, I'll take you to the beginning of my story. So we always watch the news, and the news is always full, unfortunately, of negative events and bad things that are happening around us. Rarely we find news that talk about positive things. And maybe this is a feature of, we always like, the notorious, the sensational. But, from watching the news, that's one place where I see what everybody sees and think what nobody has thought. As a human and as a scientist. And so with the start of the Syrian crisis and the influx of Syrians across the border, I do not like to use the word refugee, because it's a term coined by the West. And to me, the Syrians who came over are actually family and friends. And I'm half Syrian myself and half Palestinian, with a history of quote unquote refugees. And so to me, when they first came to Jordan, people opened their houses and homes as family and friends. And it was then later when international NGOs, with all good intention, wanting to come in and help them, that they became grouped under the term refugee. And unfortunately, overnight, they became the other group. So we really have to be careful what words we use and how that shapes our perception of the world going forward, and how can we draw from our own culture to choose the right term, the right word to describe the world around us. So with the explosion of the crisis, I mean, the Syrian refugee crisis being one of the biggest, not just in this century, but even in the past, the world has been, the numbers are exponential, as you can see. Where the numbers of refugees and then where they are, and as you can see, they're all situated actually in the Middle and East and the closer Far East. I don't know, Pakistan is considered part of the Middle. I mean, that's another discussion. But there's a lot, unfortunately. And they're all living not in camps, but actually in urban settings. So they're part of the community, of the local community, as you can see here, whether it's rural or urban. And so with that in mind as a history and the Syrian crisis unfolding, we realize that many of those Syrian refugees are actually children. And that is something that has to capture our attention because not just that 52% are under the age of 18, as you can see here, there's also multiple displacement. So this is a crisis that does not appear to end. And the consequences on them and the surrounding community is profound, in the sense not just in terms of food and nutrition and shelter, but I want to go deeper and talk about other let's say issues and sectors that have not been addressed in the past, which are mental health issues. So 45% have PTSD, 20% are clinically diagnosable with levels of depression, and we could go on, right? And again, many of them are children. And so this is a very important issue that we want to shed light upon as we develop solutions and programs and interventions to deal with the crisis, right? We know food, basic health, and shelter are always addressed, but it's the other things that we want to address. And so therefore, we wanted to evaluate as scientists the interventions that are being deployed among refugees for various purposes, whether it's to abate violence or promote health. And we want to see how can we really do a robust evaluation for such programs, and how can that robust evaluation actually help, and how do you actually do it, right? So we want to know what works for whom. So we want to look at quality and depth of the evidence regarding humanitarian public health programming globally. So there was a funder, R2HC, which was willing to fund such research. Usually funders don't go into those sectors because it's very difficult to conduct proper quality research, randomized control trials, etc., under humanitarian conditions. So they were interested to fund us as a group of scientists globally to study the impact of an intervention that was being run by an international organization within Jordan on Syrian and Jordanian youth. So this is a program called No Lost Generation, and it's an 8-week psychosocial intervention that actually trains these youth ages 14 to 18 on either weaving or haircuts or computer or sports and things like that. And the whole program was about boosting, it was psychosocial, but it was about alleviating toxic stress and building resilience. So that's what the program actually promoted or claimed, and to support and heal conflict. Now, what we innovated in this scientific project or scientific practice was to boost the independence and implementation of the research teams. How do we do randomized control trials? And what were the methods we used to go beyond self-reports? So I'm going to highlight these 3 points because, as I said, this is a story, a journey on how where we are today and how we got to epigenetics. So we first wanted to make sure that the team that was doing the evaluation was truly independent from the implementing team of the program itself. So we had we had Yale University, we had Queen Margaret University from the UK, we had also Harvard University and University of Western Ontario from Canada. So we had a group of scientists, and it was led by us in Jordan through the Hashemite University and Tahrir organization to lead the evaluation of the program that was implemented by Mercy Corps. And we emphasized this very much because we wanted to ensure that the participants would be able to give us neutral feedback and not related to Mercy Corps. The second thing was, how do we do randomized control trials? Again, this is a humanitarian condition. How do you choose who to give the intervention, who you do not give the intervention, while maintaining a high level of ethics? So what we did is, there was funding only for a limited number of participants. So we actually gathered the names of the children who were eligible for the program, and then we allowed them to draw lollipops from a bag that had different colors. And then we would flip a coin at the end of each day, assigning a color to who would be assigned control and who would be assigned as the experimental. And every time we would flip a new coin, so nobody could, say that one color was always going to be the experimental and one color was always going to be the control. So it was a fun way of doing things, and it was done in collaboration with the research team on the ground. And again, I'm going to talk about the research team, which was the Jordan Syrian research team. So we actually recruited research assistants from Jordanian and Syrians to go and gather the data, and not just only gather the data, but also participate in the design of the research, in the evaluation of the surveys that we were going to implement, in the translation of the surveys to make sure that they were applicable and culturally relevant to the community, and then in understanding and analyzing the data as it came through. So it wasn't just an independent group of scientists from outside internationally. It was a true partnership of working together to try to understand how to do better science to really understand how the evaluation works and how to do it better. So we had almost 500 Syrian refugees and almost 400 Jordanians, as I said, between the ages of 14 and 18. And we did 3 ways. So we did a baseline before the intervention. We did 2 months after the intervention ended, and then 11 months later. So this was a longitudinal study. And we wanted to look at psychosocial markers, biomarkers, cognitive function, and genetics. So we started with self-reporting and looking at surveys at these 3 time points. And this was more self-reporting, right, by the Jordan and Syrian research assistants working with the kids. Now, this is the standard way of doing things. However, we all know that a child may not respond, or even any participant may not respond in a way that would really reflect what's going on inside. However, we did this for due diligence and to have it as a base that we could fall back to and compare. And I'm telling you the whole story to tell you that you cannot isolate and study one thing without studying the whole human being in a holistic fashion, because we are not just little factors. We are a combination of different things that is going on in our body. So we had a battery of surveys that were adapted for the Arab culture, and that we also critically, we meaning my team, the research team in Jordan, critically addressed to make sure that it was relevant and it made sense. And as you can see, we did the evaluation and we found some of them actually showed that there was a change and some of them did not. And this is typical. If all the surveys showed us that the intervention worked, we would be skeptical. And if they all didn't, we also would be skeptical. But when you have some that showed that there was improvement in certain areas and some that didn't, then you can now start trusting that this intervention, that the research was done properly. And we published the results of this part of the study as in the Journal of Child Psychology and Psychiatry. And we showed that insecurity and distress were reduced. However, one interesting thing is that as we were doing the research, as you can see with the different surveys, my research team, as they were doing the evaluation with the kids, realized that some of the children, after taking these really stressful surveys, felt really depressed from taking the survey, because it was asking about, how do you deal with trauma and how are you feeling and so on. So my team actually informed us that, can we end on a more positive tone? Can we ask something that will let them leave this 1 hour of intense survey and data collection in a more positive way? And that's what led us actually to develop a resilience scale that we deployed among these children, as you can see. And so this was the beginning of another part of the story that was actually driven by the local researchers, because they're local, they're Jordanian themselves, they're Syrian themselves. So they understood what the kids were going through. It wasn't the other group. It was all of us in this together. And because of that, we developed this resilience tool and actually measured the resilience of this program, whether it really improved resilience or not. Now, the program had claimed that it does improve resilience, and what we found that it actually didn't. And our work was not just published in Child Development, but actually was featured in Science when it came out. And so talking about looking at positive outputs, not just only negative outputs, this is a very important point. In research, we're always looking for the negative, especially when we're dealing with stress and trauma. We don't look for the positive. Yet through evolution, we have evolved and we have survived. So I'm sure there are positive things, and we want to shed a lens on that. The second thing is that driven by the local people to ask the right questions. And thirdly, this program, No Lost Generation, that was run by Mercy Corps, claimed to boost resilience. And when we tried to dig deeper to understand how come we could not see any improvement in resilience, we found out that it was because the program was actually designed by the West, in the West, for another culture, right? The Jordan and the Syrian. And they had focused, the Western culture focuses when you build resilience on the individual, while our communities are more focused on the community and the family. And so the intervention did not address family and community. And we showed this in our study, that the resilience measure showed that if you only focus on individual, this program is not working. And so the learning from this was that Mercy Corps went back and changed their intervention to include a component that actually included the family and the community to be able to really improve resilience among these kids. The other, but again, surveys is the traditional go-to, and that doesn't necessarily translate really what's happening in the body. So we also wanted to look at cognitive function and how does the stress affect the brain. So we actually, the kids played computer games to assess those cognitive functions. And again, we were very surprised that we found that the trauma and war and conflict did not actually have any impact on cognition. The biggest impact came from poverty. So if you were a Syrian or Jordanian exposed to trauma or not, the really thing that affected your cognitive ability was if you were poor or not. And this is a very important message to WHO and others, that we need to focus on poverty and alleviating poverty if we really need to help children around the world. Now, then we went on, we wanted to see the stress under the skin, what's really happening in your body. So we wanted to look at biomarkers, right? And track them over time. And we wanted to do it in a way that was culturally acceptable. Of course, Western scientists were skeptical. How can we really use this? How are people gonna, how the children or the family is going to accept it? And Mercy Corps themselves, who are implementing the intervention, were very skeptical about measuring biomarkers. But this is when we stepped in as locals, right? From the community, from the refugee community, saying, no, you are skeptical because you are from outside the community. So you feel people will not trust you or not understand why you're doing it, or there'll be a misunderstanding. But we are from the community. And so the approach that I did, I went down and talked to the elders of the community, the parents of the children, and the children, me and my team. And we explained that, we want to evaluate this intervention that's being implemented on us, meaning you, them and us, the children, the parents, and us as scientists coming from the community. And we wanted to see, is this intervention that's designed by this international NGO, does it really work? So it was about giving agency to the parents, to the refugees and the Jordanians, giving them agency to evaluate the program and then explaining the science of how to do it. And first of all, this raised awareness among the community to the science, gave them agency so they're ready to participate, and also to inform how to do this research, how to do it better, how to understand the results that are coming out. So it was a fantastic collaboration and partnership between the community and between us as scientists that we then conveyed to the rest of the scientific group. So everybody was excited. So, for example, we talked about, that the surveys themselves are not enough, that we need to go under the skin. So we need to look at the biomarkers, the hormones and the the inflammatory biomarkers that reflect our bodies. So we want to look at inflammation, and then cell-mediated immunocompetency and cortisol secretion as an indicator of stress, right? And because we know that there's an association between poor health outcomes and life course of an individual. So for inflammation, we looked at CRP as a biomarker of inflammation. CRP stands for C-reactive protein, and we can measure it from a blood spot. So and there's a lot of evidence to associate adversity and inflammation, especially in adolescents. And same thing with cell-mediated immune function. We wanted to look at EBV, the Epstein-Barr virus, which is a herpes type of virus, which also has an association with stress and chronic stressors. And so what we did is we went to the parents and the children. We said, hey, if we really want to understand if the stress and trauma that you're going through really is affecting you, we need to look at these things. And the only way is to do it by looking at blood. What do you think? And they said, yeah, but if you're going to take a lot of blood, nobody's going to accept. And we start thinking, oh, okay, so let's look at science. And science says, oh, it's, technology is advanced. All you need is a little drop. which is like what you do, that little little needle that you test when you have diabetes. And because unfortunately diabetes is a global epidemic, everybody has diabetes, taking that little needle to test your sugar is something everybody's used to, and everybody said, oh, we can do this, don't worry. So everybody was on board and actually helping us how to talk to people and how to address it. And they were helping each other to figure out ways of doing it. And then, so we did the blood. And then the last biomarker that we were interested in was cortisol. Now, in order to do cortisol, you can't do cortisol from a drop of blood. And so we said, we got to look at other stuff. And of course, this is a conversation with the families. And we said, well, you could take, unless you took a lot of blood, which is not easy. So we said, you could take saliva. But then imagine spitting into a tube for a long time and it's messy. You want to make sure that children have not eaten at least a half an hour before. So then we told them, but you could find cortisol in the hair. And if you take a sample of hair, just as a few strands, like 100 strands, which is not more than the top of a pencil, then the job is done. But I said, could you really do that? Would people, agree to taking a sample of hair? Oh, all the parents and the kids were excited. They said, oh, we'll do haircuts. We'll get barbers and salons, and we'll give everybody a haircut, and we can take that sample of hair, and we'll understand the science behind it. So the international NGO was skeptical about doing that. We had all the community excited about the science doing it. And why cortisol? Because cortisol is a stress hormone, and it reflects how much stress you are. And as you can see, when you have a trauma, it goes up, and then it goes down. And then if you have more trauma, it goes up. And then if you're really hyper traumatized, it could really go down. So it's a really good indicator of stress. And so we collected the blood spots, we collected the hair, and we were able to ship them to do the analysis in the appropriate labs. And so we were able, under humanitarian conditions, to actually do stress biomarkers and evaluate this intervention. So to share some results for the biomarkers, for the CRP, actually, we did not see any change with time with the intervention. And of course, again, not everything that we chose is going to work. And this is in itself a learning. We also did not see any difference with the Epstein-Barr virus. However, we did see a change in cortisol. So we showed that for those individuals who had high cortisol, meaning very stressed, it went down. For those who had very low cortisol, meaning they were hyper stressed, it actually went up. So there was some kind of normalization, which spoke to the effectiveness of the intervention for alleviating stress by showing, as you can see in a summary, that cortisol actually went down after the intervention, for both Syrians and Jordanians. And this was research done by different members of the research team that I mentioned from different universities. Now, we also wanted to look at genetics, if you remember. So that was the fourth component. And this is where the story is leading us. And why did we want to look at genetics? Because, we are not just shaped by our DNA and not just shaped by the environment. We're shaped by both and the interplay of both in our bodies. So in order to understand that, we wanted to understand what is that interplay between the environment and genetics. Now, how does that really happen at the level of the cell? So cells in our body actually listen to what's happening outside. Not literally listen, but it's through signaling transduction. Signals bind to receptors, and then receptors convey those signals within signaling transduction cascades into the body, the body of the cell. And the cell could either respond in short term by mobilizing enzymes or movement of the cell and so on, but also on long term by affecting gene regulation at the level of the DNA. So either turning on or turning off genes. And that's what epigenetics is about. Epigenetics is how the cell responds to the environment by turning on or off genes. And I'll give you a simple example of epigenetics and how it started with mice. So we all know that if you have two mice, one that is stressed and therefore does not take care of its pup, and one that is not stressed and takes care of its pup. And mice, when they take care of their pups, they lick them, right? They take care of them. And so, and what we showed is that the pup that was not taken care of by their mother because it's low nurturing grew up to become an anxious adult mouse. And the pup that was taken care of by a high nurturing mother grew up to be a more relaxed adult. And these are identical mice in terms of genetics, right? So then this difference of the pup growing up to be anxious or relaxed was as a result of the environment, meaning the caring of the mother. So we wanted to understand that more. How does that happen? And whether it can be transferred from generation to generation, and whether it can be reversed, right? So what we found is that you can actually reverse the impact of this low nurturing mother on her pup by injecting a drug that adds methyl tags to the genes that were unmethylated. And you go from a relaxed, high nurtured rat to an anxious rat just by changing the methylation, which results in turning on or turning off of genes. So what was that gene that was being turned on and off in these anxious rats? So we know that the glucocorticoid receptor, which actually helps shut down the stress response, is a gene, right, that binds cortisol. So when you have cortisol, it's secreted in your blood when you're stressed. Now, what happens is that you have receptors, the glucocorticoid receptor, that binds cortisol and reduces it in your blood, therefore reducing the stress. Now, if the gene for the glucocorticoid receptor is turned off by the environment because of what's happening, a pup is being licked or not licked, being nurtured or not nurtured, then you don't have enough of that receptor, and therefore the cortisol hormones remains high, and you grow up to be an anxious pup. Again, if you inject a drug that changes the methylation tags, you can turn on the gene again, and now you have glucocorticoid receptor expressed, and now you can reduce the cortisol. The question is, how come through evolution we still have anxious behavior, right? Why did this response, this epigenetic response, remain? You would think that if you're anxious and cortisol goes up because of the trauma around you, that this should be, eventually removed because it creates anxious pups or anxious adult mice. But we know being anxious has a survival advantage, because then you're more vigilant, you're taking care of what's happening, and you're not just relaxed and you don't care what's happening in the world. So there is an advantage to have this anxious behavior on the long run. However, again, just to keep you in mind, that we're also always asking the negative questions and we're focusing on anxious behavior. Nobody has asked the flip question: what are the positive behaviors that have been selected for also by surviving a trauma? Nobody has asked those questions because, again, people love the notorious news. So we'll get back to that in a moment. So moving to humans, I mean, a lot of research has been done on epigenetics in mice, and there are many, many examples, or other animals. However, in humans, if we want to follow the trajectory of my story, looking at glucocorticoid receptors and cortisol and stress, and how does that help or result in anxious adults, we would have to look at the brain. And of course, for ethical reasons, we cannot go in and dissect a living person's brain and look at it, but we can look retrospectively, and we see that that there are changes in epigenetic tags on certain genes that affect behavior, and that these can be or have been associated with experiences from the environment of the child growing up. And so we, in our research project on the Syrian and Jordan youth within this program, we wanted to look at something related to genetics. We wanted to see, taking epigenetics as our background, whether there are genetic variants that would predispose children to be more anxious or less anxious, or more stressed or less stressed because of the traumatic exposure that they have been exposed to. So we want to test to see if there's associations, and we want also because that increase or decrease because of the intervention. So we want to check both. What is the impact of the trauma, and then whether the intervention can make a difference. And because there has been evidence to that effect in the past. So and of course, there's environmental sensitivity among people, right? Some are more sensitive to trauma, some are more sensitive to interventions, and some are less sensitive. So we wanted to understand, what does that mean at the level of genetics? And the only way is to do the experiments. And we had this opportunity to look at this large sample number, and we had gathered a holistic kind of picture of these children through the other tools that we had used, the surveys, the biomarkers, and the cognitive function. So now we can ask the genetic question in a more real way. And this is very important. And that's why I had to tell the whole story to get to where we are now. So we wanted to look at, first of all, genes that already have been reported. So two we wanted to focus on were MAOA and COMT. And because they were involved in neurotransmitters that had to do with behavior. And we focused on MAOA as a neurotransmitter because it actually inactivates dopamine. Okay, and we know dopamine has to do with behavior, especially under stress. This is an X-linked gene, so only males have only one copy, and that's where you can actually see its impact on behavior. And the interesting thing for this particular gene is that it has repeat polymorphisms. It has both high and low repeats, and so it's like a toggle for high activity and low activity variants. So we wanted to look at these children and see who had a high activity variant and who had low activity variants, depending on the number of repeats, and see if there's any correlation with their exposure to trauma and their resilience, for that matter, and how they perceived stress and the trauma they were exposed to. Because there has been research in the past indicating that MAOA has an association between behavior and early life stress. And what we found is that there was an association between MAOA with perceived stress. So some individuals had low activity and high activity, as you can see in this graph, and that we showed that those who had a higher perception of stress, it became reduced with time, but it became reduced more for those who had the low activity of, became reduced more for those who had a high activity of MAOA. And this is without just showing the clear lines. So lower activity individuals had lower perceived stress. And then, and this was highly statistically significant. And then we wanted to look at whether there was a correlation with resilience. So we're looking at 3 dimensions. So as you can see here more clearly, that resilience had an additive effect with MAOA on perceived stress. So so MAOA was still associated with perceived stress, and resilience had an additive effect, as you can see here. And so this was very interesting and correlated with what research has shown before, and illustrated the influence of the gene on resilience, right? And this is very important because, again, we're looking at a positive output now, not a negative one. We're not looking just at perceived stress, we're looking at resilience. And to us, this is what we want to call for as we develop new research projects, to look at things in a different way, in a positive way. And as you can see, we were able to follow this longitudinally because we had the 3 time points, and this was published in PLOS ONE for those who want to learn more about the results of the research. But this led us to this environmental sensitivity that I mentioned earlier. And I just want to propose this, that so as a result of the interplay between the environment and genetics, we can categorize people either as orchids, meaning they're highly sensitive and have to have the right environment to thrive, otherwise they'll wither. And then those who are dandelions, who are more tough, and therefore they can adapt to any situation wherever they are. And then we have the tulips who will fall somewhere in the middle. Not too delicate like orchids and not too strong as dandelions. And so we need to have that perspective in mind as we study the responses to interventions by different individuals, that it's not just a black and white, it's a much more complicated picture. And for that part of the research that I had shared, these are our different participants and partners as we go forward. Now, so this leads me to the last part of my talk, which is, again, back to the first sentence, seeing what everybody sees, but thinking what no one has thought. So doing all this research, it struck me, being half Syrian also myself, and knowing the history and the culture, and being a molecular biologist, can these epigenetic signatures that we have just been talking about and newly discovered and people are working on, can they be transferred across generations? I mean, I'm not the first one to ask that question. It has been asked in the past, and we already know that it happens in animals, right? In mammals. And even in primates, but does it happen in humans? That's the fundamental question. In mammals, we know 1% of genes escape the epigenetic reprogramming, and that signature that happened in the lifetime of the adult is transferred to the zygote and therefore to the embryo. But that 1%, what are those genes and how does that impact our behavior? Is a question that we don't really understand in humans yet. For ethical reasons, we cannot actually do those experiments that we do in mice. So then it goes back to, are there experiments that happened serendipitously by those around us. And so knowing my culture and my history of the region, I saw an opportunity to learn more in order to help humans and humanity in the future, because we know that so we wanted to ask this question, that if the mother is pregnant and she was exposed to some kind of trauma, in this picture she's exposed to smoking, but I'm going beyond the trauma of war, will that elicit not just an epigenetic signature in her, but in the fetus inside her? And if she's carrying a female, and we know that a female during embryo development will already have the eggs developed, will those eggs also be impacted by that trauma? And will that be a direct exposure to the trauma by virtue of being a living cell when the trauma happened? Or is this an epigenetic signature that is being transferred from the mother to the daughter to the granddaughter? And in order to ask that question ethically, looking at trauma, not smoking, as the let's say as the factor impacting the epigenetic signature, again, we cannot do that. We cannot design an experiment like that. But I already know of the history of the culture, that in Syria there's a city in Hama that was exposed to a huge trauma by the Syrian regime, and more than 20,000 people were tortured and killed and imprisoned. So we were able to go back into those families and study and capture samples from grandmothers who were pregnant then, their daughters and their granddaughters. And we were able to create an elaborate, elegant experiment comparing grandmothers exposed, mothers exposed, and then controls where nobody was exposed, to actually ask that question. Can the epigenetic, is there an epigenetic signature of trauma, and can it be transferred across generations? And what does that look like? What are those genes? And my daughter, of course, who, I involve my family in everything I do. And because this is a family project related to family members as well, we called it the immortal kiss of our grandparents in a positive way. And as we started this study, it wasn't just about what are the negative impacts of the trauma. We also wanted to look at resilience and what are the positive impacts of this. And we are actually working on this. We finished collecting the data, and we're actually in the middle now of analyzing the research. So stay tuned. This is also my collaborators from Yale and Florida, Connie Mulligan and Catherine Panterbrick, as well as all the Syrian team and the Jordanian team. A movie was done about our work, and I'm ending now, which was called Terror and Hope, to bring to the global community around the world how the science of resilience is the name of the movie, how we can understand what's happening on the ground so that we can, support science to do better science when we involve everybody in the science and give back. So we gave back all the results back to the community. We shared it with the kids and their families so that the circle of agency can be complete. So I'll stop here with a few sayings by Mahatma Gandhi and Nelson Mandela, that if we are to teach real peace in this world, and if we are to carry on a real war against war, we shall have to begin with the children. And that's the essence of what we're doing. And if we are to evaluate the society's soul, look at the way in which it treats children, and education is key. So again, a thank you to all my team over the years for the different research projects that I have, showcased to you with a story. And I'll stop here, and I'm ready for questions. Thank you very much.
Thank you very much, Dr. Rana, for a wonderful presentation. And we are receiving a lot of messages regarding your presentation, and people are really fascinated by your work. And they really appreciate all the visuals that you have presented in the presentation. With this, let me read out some questions for you. So we have first question. Okay. Yeah. Please give me a minute. I think I have lost the question. Okay, yeah. So we have question from Ms. Nada Hamza. She's asking, what other biomarkers can be used other than the stress one, be used for these kind of studies? And number two is she's asking about the sample number for these studies.
Oh, yeah, and I'm assuming she's asking, because again, I talked about a lot of stuff, right? If you're talking about epigenetics, I mean, in terms of numbers, the more the better, right? The more the merrier, of course. But because epigenetics is very new, any number is fine if you're if you're finding a change or a difference, or you're discovering something new. However, one way to go is to look at the literature, right, for the field, because what are you studying for epigenetics? Are you studying, what's the question? And then look at similar articles that have addressed similar questions in that field and see what numbers they have used. That will give you a gauge of the ballpark of the sample number that is expected and approved in general by the field. In terms of what other biomarkers, again, it depends on your question. What are you looking at? Are you looking at stress behavior? Are you looking at resilience? Are you looking at leadership and confidence? Are you looking at agency? So different behaviors have different biomarkers. Again, all this is a very nascent field. This is very new because it's very interdisciplinary, going from behavior and psychology to biology, and how we still don't understand this, and we're trying to make those connections. However, again, you would have to go to the literature and study what biomarkers have been identified. And I'll give you an example. So we've gone beyond, this is, so because I mentioned the Wheel of Reading program, so now we're actually working with Michael Kober's lab in British Columbia, Vancouver, and we're studying the impact of Wheel of Reading as a reading program on children and their mothers, and looking at the interplay of the dyad, looking at epigenetics. So we're just taking cheek swabs and looking at the epigenetic signature. So again, that is in a way a kind of a biomarker. But if you want to look, we're looking at also, for example, leadership qualities. We're looking at hormones, mean sex hormones, and what's related to the menstrual cycle, because that has something to do with your how you feel and how stressed you are. And those we collect them from blood. So again, depending on your question, you can look what are those biomarkers that are related to that physiology and behavior, and then you can start looking what sample I have to take to find those biomarkers, and where the technology has advanced to help you figure out what type of samples that are not very intrusive that you can use.
Thank you very much, Dr. Rana. And so here is another question from Ms. Arada from Azerbaijan. She's saying, Dr. Rana, thank you for the interesting presentation. My question is about epigenetics. How traumatic situations coming from not your parents, but from the previous generations can affect cells and behavior?
Yeah, that's the question that we're all asking, right? Can the grandparents exposure affect their DNA, meaning turning on and off genes, and can that be transferred across generations? That's the question that we're trying to understand in humans. We know it works in mice, for example. For humans, the research that's been published, to my knowledge, of course, every day there's something new, is that there have been associations in general. That I know the grandfathers were exposed to war. Now you see that all the grandchildren have diabetes or are stressed or have a particular illness. However, to really, for it to be causal, right, you need to understand the mechanism, the molecular mechanism. Nobody's been able to address that question in particular. And we hope through our work that we can actually kind of tease that out because of the different groups that we've been able to create and find retrospectively, as I said, because of our intricate knowledge of the culture, and try to start addressing that question.
Thank you very much, Dr. Rana. So we have another question. Let me read it out for you. There are so many comments about your fascinating work. People are commenting on your, fantastic use of science for the intervention for the communities. So here's another question. I don't know how related it is to your talk. My question is whether early whitening of hair is related to stress or not, and how can we determine its impact genetically?
Yeah, that's an interesting question. And well, it probably has to do with stress at the level of the cell, right? Because it's all about oxidation reduction and antioxidants in your cell and how those are formed. Because of the the aging of the cell and the production oxygen radicals. So it is related in that sense. But again, to causal relations and tie them, whether that's transferred or not, but within the cell, definitely there is evidence for that. I hope I answered your question. But again, remember, we are all specialized scientists. We know better in our field more than other fields. So whitening of hair is a different category. I mentioned hair as a way to get cortisol, but to understand what are the intricate details of the whitening of hair, I leave it to the expert humbly, because I believe everybody knows better in their field. But I'm sure there's a lot of literature you can go read about.
Yeah, talking about hair, we have a comment from our participant. She's saying, we women, when we go through difficult times, we cut our hair short as a common practice. Does this does this release stress?
Again, I don't know unless somebody has done experiments to correlate and to answer that question. But just off the top, I mean, if you cut your hair, you feel better yourself. So it's a more you took action, you had agency, or because you heard somebody tell you that, and they told you, if you do this, you will feel better. So psychologically, you do this and you say, I'm gonna feel better. So it's like the placebo effect, right? And that speaks to the power of the brain and the mind that we still don't understand. How does that happen? And this is a whole new field of research that I encourage you all to think about. You know, you're all smart people and you all have a different DNA. So you are unique and you have a unique perspective on life and the world around you. So ask those questions and follow and pursue your passions. And maybe you will answer those questions and share the results with us and help push the field of science forward to help each other.
So, Dr. Rana, I would take the last two questions. This is from Mr. Ghulam Murtaza. He's asking, epigenetic traits are fixed in any family, or it may affect any of the trait in the coming generation? This is his first question. And his other question is, how we identify in new generations that modified trait are due to epigenetic?
Can you say the second question again?
Yes. How we identify in new generations that the modified trait are due to epigenetic changes. That's what he meant.
Yeah, I mean, to answer both questions, we don't know a lot about what traits will be inherited and changed or not. These are all questions we don't have the answers to, and again, the only way to answer them is to have a causal mechanism, molecular mechanism, to say A goes to B to C to D. Otherwise, they're only associations, right, at this point. And we have to be very careful when we talk about associations, because the famous example I always tell my students is that if you cut the legs, if you tell a frog to jump, it jumps. If you cut its legs and you tell it to jump, it doesn't jump. Your conclusion is, if you cut the legs of a frog, it stops listening. Okay, now that's a wrong conclusion. But if you don't know, you don't know, right? So you have to be very careful and not just jump to conclusions because of an association that you saw. Therefore, to answer that question, we don't know unless we figured out the causal, the actual causal mechanism, molecular mechanism. And that's why we need more researchers, more scientists working on this, so that we can start unfolding and understanding who we are as humans, how who we are is a product of the environment and our genes, the interplay between them. And then that interplay, how much of that are we transferring to the next generation? And how is it transferred? And how can we reverse it, right? That's what we want to know. If there's something negative, can we reverse it? If it's all just chemical? And if there's something we can improve and make better as well. But bottom line, as we understand and answer these questions as scientists, bottom line, it's all about being positive, having a high ethical values, taking care of each other, which are universal values that we all have, that have evolved with us over millennia, and that have been fundamental to our survival as a species. And so sticking to those, we can't go wrong, but it's nice to understand how they work inside our bodies.
Dr. Rana, if you allow me, I would like to take your quick comments on some of the recent questions posted in the chat box. Should I ask the questions?
Yeah, go ahead.
Yeah.
I'm all yours.
Thank you very much. Can these trauma-related traits be alleviated by crossover between people from different families, like simply marry people outside your family and reduce the stress?
Again, we don't know for sure. We know there's associations, right? We don't know what's happening. They could be very positive things. Again, let's not just think negative, positive. And it's not just one. Again, we are holistic. We're not just a product of one thing. We're a product of millions of genes and billions of interactions. So there's not just one thing. But we all know, even from our culture and heritage and our religion, to marry outside, we know recessive genes will come together. So we need to do more to increase the diversity. Diversity is always good. We know that, right? So whatever kind of diversity is good.
Right. So here is another question. Do identical twins have same epigenome?
Yeah, that's a great question. And there have been studies on identical twins to really understand more of the epigenetics. And yes, they have different epigenome because yes, their DNA is identical because they came from the same zygote that split into two. However, the environment they were exposed to is not identical. Even if they grew up in the same house, it's slightly different. And therefore their epigenetic signature is different.
Thank you, Dr. Rana. So here is the last question. Are autoimmune diseases a result of genetics or epigenetics? How are they related?
What disease?
Autoimmune diseases.
Oh, again, I want to reiterate, epigenetics is a very new field. We still don't know a lot. So such a very broad question is I cannot answer like are autoimmune disease related to epigenetics or genetics? It could be, yes, it could be, because again, we know that many diseases we don't understand the reason for. We look at the genetics, we don't see anything. We try to understand the environment, we don't see anything. And that we call them complex disorders, right? Diseases, because they're really multifactorial. Epigenetics could be one part of the puzzle, right? That we're now discovering. So maybe.
Thank you very much, Dr. Rana. And with this, you can see the comments. People are asking if it's possible that they would like to have your presentation. If that's possible, you can send it to me and I can definitely put it through to our participants.
Yes, absolutely. And this is recorded. So probably the whole talk will be also online, right? Available for everybody to watch as well.
Yes.
Yeah. And anybody can contact me. My email was in the last slide and you can share that. And thank you very much.
Thank you very much, Dr. Rana, for your time and
fascinating work. Kindly allow me, may I ask, it is not a question, rather a point. Dr. Rana, thank you so much for a wonderful lecture. I still remember our first interaction at ICCBS. We talk about storytelling and I'm still interested. So I was just wondering that can we initiate some sort of program under the comstech umbrella about this We Love Reading? Can we have some sort of program in which we involve children from OIC region, from Pakistan and other countries, so that they can come up online and we can involve more and more children in reading programs? Because I can talk about Pakistan, our children are not habitual of reading at all. So it would be a wonderful initiative if we can talk about some program like that.
Absolutely. And you're not alone. It's not about Pakistan. It's the whole world.
Yeah,
children don't read for fun. So it has nothing to do with global north, south, socioeconomic status. Everybody needs this. We are ready to work with you. We actually put We Love Reading now it's all online for free for anybody. So it doesn't have any cost, and I'm ready to work with you so we can reach more children. And then if anybody wants to do science on it, like we're now doing, everyone is welcome as well. So we would be honored to work with you to spread the program to reach more and more children. by
Yeah.
translating the training into the different local languages and putting them online, which are very simple, or even doing it offline.
Mhm.
We are ready to work in any possible way.
Excellent. So I think Hazima, me and you can put up some concept note, and then we can discuss with His Excellency Professor Chaudhary and Dr. Rana, and then definitely we can put up this program under comstech umbrella.
Yes, I think that's a fantastic idea.
Thank
Again,
you.
thank you very much, Dr. Rana, for your time. And I would also like to thank our participants who joined us and remained with us for the whole of the lecture. And with this, I would like to tell our participants that this lecture is recorded, and I have shared the YouTube link in the chat box as well. And you can also go to Comstech's YouTube channel and see this lecture once again if you would like to. And thank you very much, Dr. Rana, for your time. And thank you very much to all our participants for joining. Hope to see you sometime again, inshallah. Bye-bye.
Assalamu alaikum. Thank you very much.
Assalamu alaikum.
Bye.